Hello, my name is Drew, and I'm going to be covering how influenza causes damage in cells for my learning object for assignment. The flu virus is covered by a lipid envelope, classifying it as an enveloped virus. Attached to the lipid envelope are little blue and red projections on the image I've drawn. The red spikes are hemagglutinin, and the blue spikes are neuraminidase. Hemagglutinin is responsible for recognizing and attaching the virus to host cells, while neuraminidase initiates the infection by attempting to enter the host cell. Inside the envelope, there is a capsid cover, and inside of that, there's a bundle of RNA. One of the main processes that influenza uses to damage cells does not even originate from the virus itself. Instead, cytokines belonging to the host's immune response initiate a deliberate suicide, otherwise known as programmed cell death. This limits the virus spread by purposely killing off infected cells. Here, cytokines activate death ligands, seen in red, which in turn activates FADD. FADD activates caspase 8, seen in yellow. Caspase 8 activates BID, which forces the cell's mitochondria to produce cytochrome C, a vital checkpoint needed to produce apoptosome. Caspase 8 eventually interacts with caspase 9, originating from apoptosome, to activate DNase, which fragments the cell's DNA, destroying the host cell. Of course, the flu virus can induce damage itself. A major way in which the virus kills off cells is simply that the rapid replication disturbs cellular function, damaging tissue and accumulating more and more virus particles to do the same thing. Additionally, the virus can initiate cell death using the host's own processes intrinsically or extrinsically. Here, the intrinsic pathway is shown. The virus activates VDAC1, ANT3, and the Bax-Back complex to induce an outflux of cytochrome C into the cytoplasm. However, the cell has mechanisms in place which serve to stop this process from continuing, like the apoptotic inhibiting protein API5 and the Bax-Back inhibiting protein RFN43. Both of these are inhibited by the flu virus upon entrance into the cell. As a result, apoptosome remains uninhibited and the cell dies. Lastly, the extrinsic pathway resembles the cytokine-induced apoptosis. Like cytokines, the virus increases the expression of death ligands FASL and TRAIL. These two activate FADD, which carries on the process seen in the cytokine slide. However, again, the cell has mechanisms to protect against this. In this situation, however, the inhibitor is FLIP, whose concentration is decreased by the virus. As a result, FADD is uninhibited, eventually resulting in apoptosis, and the cell dies.